Okay, so we touched on chemo versus autotrophs and all that. Let's talk about what the environment itself has to be like for the growth of a microbe. Well, actually, the growth of any living thing on this planet. We're going to find one of the main environmental factors, besides the presence of water, is temperature. Okay? Goldilocks was right. Too hot, too cold, just right. When it comes to temperatures, too hot means that, well, your proteins and stuff are going to denature. Your plasma membrane is going to be too fluid and thus leaky. Too cold means your plasma membrane is going to be too stiff. Things are not going to be able to diffuse through oxygen, water, and some small nutrients. Also means your enzymes are going to work slower if it's too cold. So what you want to have is just right. Temperature is warm enough so everything's fluid and moving, but still cool enough that it's not moving too fast, not falling apart. You look at this figure from the textbook. You'll notice that we've split microbes into, 5 groups, 5 generalized groupings. To the far left, these are going to be the psychrophiles. These are going to be the things that want to grow in the cold. Yeah, there's microbes growing in the snow. When you get to high elevations or you get to the north of the South Pole. If you look at the temperature range there, optimum for them is going to be 0 to 5 degrees Celsius. It's going to be around 40 degrees Fahrenheit for those of us who think imperial. This is basically refrigerator freezer temps. These are the microbes you're going to find that are going to be in, your food, that are going to still cause food to spoil in the fridge quickly. Now, we humans, we're mesophile, the green line. We operate best 37 degrees plus or minus a degree or so. 98.6 degrees Fahrenheit. That's our body temperature. Too hot, too cold there, we fall apart. So, the interesting thing is, if you look at the psychrophiles, you look at the extreme thermophiles that are 7 degrees Celsius and above. That's like 150 degrees up, well above boiling. These are going to be the hot springs down at the bottom of the ocean, the sulfur springs. We generally don't worry too much about the psychrophiles or the extreme thermophiles when it comes to MedMicro because, well, we're either too hot for them if they're a psychrophile, or we're way too cold if they're an extreme thermophile. Think about it. You take a psychrophile, you inject it into a human, you ingest it. You're immediately going to be, 30 some degrees Celsius higher than its optimum? It's going to break down. It's going to die rather quickly. The extreme thermophiles would be like taking you and dropping you in a minus 80 degrees freezer. It's the same thing for them. They can't handle it. The temperatures are too low. Their metabolism will shut down. So for us, when it comes to actual infections, and I'm not talking ingestion. If you ingest thermophile byproducts, yeah, you can be sick. But when it comes to flat out infections, it's the psychotrophs, the mesophiles that we actually have to watch out for. The psychotrophs, we're just the edge of what's too hot for them, but it could be close. But those that are mesophiles, that like anywhere from 35 to 39 degrees Celsius, E. coli, Staphylococcus aureus, those are the ones we have to worry about.